morning to all the EAPCET aspirants. So, yes, here we are with our first chapter from plant physiology, which is very important. So, from plant physiology, we are going to cover up 5 chapters. One is transport in plants, second will be mineral nutrition, and third is photosynthesis in higher plants, fourth is respiration in plants, and fifth is plant growth and development. From all these 5 chapters, if we have to come across, we can expect 7 to 8 questions in your EAPCET exam. That important unit that this is. Okay, all the 5 chapters combinedly from plant physiology, we can expect 6, 7 or to 8 questions also sometimes. So, focus on each aspect and learn it carefully. From transport in plants, if you have to fragment this chapter and learn, first you have to focus on the transport mechanisms, okay, focus on the transport systems. Transport mechanisms, we have 2 types of transport, we have. One will be passive transport and second is active transport. Passive transport, again it will be 3 modes of passive transport we have. First will be diffusion, second facilitated diffusion, third will be osmosis. Active transport, where the transport of molecules will be from low concentration to high concentration, which is against the concentration gradient. When the transport of molecules is against the concentration gradient, it requires the energy, that is ATP. Active transport is energy requiring transport, which is from low concentration to high concentration, which is against the concentration gradient. Clear, right? Okay, both active transport and passive transport is clear to you, right? Now, after this, we will look on to long distance transport. Okay, before going to long distance transport and to understand this passive and active transport clearly, you have to know about the plant-water relations. Plant-water relations will be clear to you once you are known with plant-water relations. You will be clear with it once you are known with. First is water potential, second will be pressure potential, and third is solute potential. You should have a clear idea on water potential, pressure potential and solute potential. After which you will move on to long distance transport, where you will be exposed to unidirectional transport and multidirectional transport. Unidirectional transport will occur in xylem, multidirectional transport will occur in phloem, that is from source to sink. And in unidirectional transport, when the transport is through the xylem, you will also get exposed to apoplastic pathway and symplastic pathway, which is very important. Apoplastic pathway and symplastic pathway you will learn along with the anatomy of root from anatomy of flowering plants chapter, that is a connected concept. Okay, so this is the whole and sole gist of the chapter ma. This is how you have to go through this chapter. Every concept here in the transport in plants is important. This is easy. Mostly you will get application based questions from transport in plants. So here we go through this transport in plants first. Yeah. Plants do not have any interstitial fluid or any circulatory system. Circulatory system is absent in the plants. So how the movement of water molecules, might be minerals or organic nutrients or plant growth regulators, will go along the longer distances, that is by vascular system. Okay, that is by vascular system that is developed here in the plant body. We call them as mechanical tissues or conducting tissues. We have 2 conducting tissues. One is xylem. Xylem will look after the transport of water and mineral nutrients. Phloem will look after the transport or translocation of photosynthates, we will call it as. Photosynthates are nothing but sugar molecules. These photosynthates photosynthetes will be transported from source to sink. Photosynthetes will be transported from source to sink. Fine, right? There is no circulatory system that is present in the plant, but a strong vascular system or mechanical system is developed. Fine, right? So, this is from source to sink. Fine, right? But this is not in NCERT. This is in Telugu Academy. This is your EAPCET class, right? For your MCET, this chapter is there. For NEET, transport in plants and mineral nutrition is deleted, right, ma? For NEET though, you will have only photosynthesis in higher plants, respiration in plants and plant growth and development. Transport in plants is deleted in your NEET syllabus. This is for MCET, okay? Now, you have to know about unidirectional and multidirectional transport. Always remember, unidirectional transport will occur in xylem tissues. Okay, unidirectional transport occurs in xylem, multidirectional transport occurs in phloem. Fine, right? So, this is the transport of water and minerals in the xylem from roots to different parts of the plant, from roots to stems, roots to leaf. Whereas multidirectional transport is the transport of photosynthetes from source to sink. Remember, source is always a matured leaf because food is synthesized in a matured leaf. Okay. So, sugar for example, which is formed over here in a matured leaf, which acts as what? Source. This is source. From source to sink, different sink it is transported. It can be to some storage tissue, or to some growing parts of the plants like fruits, buds, be it auxiliary buds or terminal buds. Fine, right? Storage tissues, fruits, buds, and to younger leaves as well. Younger leaves. When different set of questions are asked to you, you should be clear. You should be clear with what is acting like source and what are sinks. Source will be a matured leaf and sink can be anything. Sink can be anything. It can be a storage tissue, it can be roots, growing fruits or buds, it can be younger leaves. Younger leaves also act as sink for their growth. Okay, they will drive out the sugar molecules or photosynthetes from the mature leaf. So, fine, right? You are clear with unidirectional transport and multidirectional transport. Multidirectional transport, here it is the transport of photosynthesis, that is organic compounds, that is glucose molecules. Here one more point to be highlighted, that in plants, if glucose immediately after formation, if glucose has to be stored, it will be stored in the form of starch. If glucose has to be stored, it will be stored in the form of starch. Fine, right? If glucose has to be transported, it will be transported in the form of sucrose. Fine, right? Starch and sucrose. Clear till here? Sometimes plant hormones and other chemical stimuli are transported in polarized or unidirectional manner. They are transported in a polarized or unidirectional manner from where they are synthesized to other parts. Remember this, plant hormones and some chemical substances, they are always transported in unidirectional manner, that is through xylem. Phloem transport is always multidirectional, that is from source to sink. I hope these 2 points are clear to everyone, right? Now we will move on to means of transport. As we have discussed, we have 2 means of transport. One is passive transport and the second one will be active transport. We have passive transport and second is active transport. Passive transport can be through diffusion, facilitated diffusion and osmosis as well. This is across the concentration gradient. Remember the statement properly. Passive transport is towards the concentration gradient, which means across the concentration gradient. Okay, just go through the diagrams here. Fine, right. This is across the concentration gradient, which is towards the concentration gradient. Fine, right. Active transport is against the concentration gradient. Fine, right. This is against the concentration gradient. Remember, across the concentration gradient means it is from high concentration to low concentration. Fine, right. Higher water potential to lower water potential. High concentration means high water potential to low water potential. Fine, right. What is water potential here? Water potential is nothing but the free movement of water. For example, if you have taken a beaker, full of water, only water molecules, the free movement or the kinetic energy of this water molecules will be maximum because there are only water molecules here. That is the high water potential. Movement of this high water potential water molecules to a beaker containing both water molecules and some solute particles. These solute particles will reduce the movement of water, or they will reduce the kinetic energy of the water molecules, which means the water potential is decreased in this beaker. Water potential is decreased. Here it is high water potential, here it will be low water potential, right? So, higher water potential to lower water potential, the molecules will be transported. This is across the concentration gradient. Against the concentration gradient, that is from low concentration to high concentration. Okay, against the concentration, which means this is from low concentration to high concentration. Low concentration to high concentration. This is against the concentration gradient from low to high. That is from lower water potential to high water potential. As it is from low water potential to high water potential, this requires energy, that is ATP. Now we will look on to the diffusion, facilitated diffusion and active transport here itself. In diffusion, if you observe clearly, diffusion takes place both for gaseous molecules and liquid molecules. This is the high concentration gradient, and that the molecules will diffuse all around. For example, if I have to spray some scented material or some scented gas here, this gas will be transported all across the room. Even if we use room fresheners, right? Room fresheners spraying at one corner of the room, the freshness or that smell will be spreaded out to the entire room, which means the gaseous molecules are diffusing all across. The same case happens in the cells as well. When the molecules, both liquid molecules might be, gaseous molecules might be mostly, they will diffuse all around the particles. That diffusion is from high concentration to low concentration across the concentration gradient. If you see facilitated diffusion is for those molecules which have hydrophilic moiety. What does this term mean? What is hydrophilic moiety? Hydrophilic moiety. Those molecules which have hydrophilic moiety, which have polar molecules like this, okay, which have hydrophilic moiety, these molecules will be transported across the concentration gradient by some carrier proteins or channel proteins. We call them as what? Carrier or channel proteins. Carrier proteins or channel proteins. fine, right, by through some carrier proteins or channel proteins. When molecules which have hydrophilic moiety means polar molecules which cannot diffuse across the membrane, they require some carrier proteins which carry them from one corner of the membrane to other corner. Okay. These carrier proteins will carry these molecules from one corner to other corner across the plasma membrane, fine, right. So, those molecules which have hydrophilic moiety, which are polar, which cannot diffuse by themselves, they will take the help of carrier proteins or channel proteins, and they will get transported from higher concentration to lower concentration, that is across the concentration gradient, which is known as what? Facilitated diffusion. The molecules transport will be facilitated by carrier proteins or channel proteins. Clear till here? Now we will look on to the active transport. Active transport is against the concentration gradient, which is from lower concentration to higher concentration. This requires ATP. This requires the energy, that is ATP, which is from low concentration to high concentrated region. Okay. This is from low concentrated region to high concentrated region. Okay. Through ATP, the molecules will be transported. Fine, right. As it is against the concentration gradient, this will require what? Energy for the molecules to get transported. This is the gist of what? Diffusion, facilitated diffusion and active transport. Now, if you look on to facilitated diffusion, as it requires carrier proteins and channel proteins for the molecules to get transported, the phenomenon, that is facilitated diffusion, is more specific when compared to diffusion. Okay. Diffusion is not specific, but facilitated diffusion is specific because it requires carrier proteins and channel proteins. The molecules are specific for these proteins. Fine, right. This is the gist of diffusion, facilitated diffusion and active transport. Now we will look on to the factors that are affecting the diffusion process. First, diffusion you see. It is a slow movement of gases, liquids, it is a slow movement of gases, liquids and solutes from high to low concentration that doesn't require any energy. It may be from one part of the cell to other part, or from cell to cell, or to short distances as well. This is not dependent on any living systems. It is only means of gaseous movement all across the plant body. This is from high concentrated region to low concentrated region. Clear till here? Now, factors that are affecting the process of diffusion. What are the factors that are affecting diffusion? Factors affecting diffusion include concentration gradient, it should be from high to low concentration, permeability of the membrane, it should be completely permeable. Temperature and pressure are dependent. Diffusion is dependent on temperature and pressure. Size of the substances, only smaller molecules will diffuse faster. Fine, right? Solubility in lipids of the membrane, and substances soluble in lipids, they will diffuse through the membrane faster. Substances whichever are soluble in lipids, they will diffuse through the membrane faster. Clear with all this, the factors that are affecting the diffusions from high to low concentration. These are the factors that are affecting the process of diffusion. Now we will go through the facilitated diffusion. As we have discussed, they require some protein channels or carrier proteins to transport the molecules across the concentration gradient, and this process is very specific, that the molecules are very much specific of each carrier proteins. Cells will select the substances for the uptake. It is sensitive, and these carrier proteins or channel proteins, they are sensitive to inhibitors that react with protein side chains. The transport rates reaches maximum, transport rate will reach maximum when all the protein transporters are used. Okay, complete protein transporters, be it carrier proteins or channel proteins, when they are used up, okay, the molecules will reach to a saturation rate, because molecules require the proteins to get transported. When proteins are saturated with all the molecules, facilitated transport will not happen. The transport rate reaches maximum once the protein transporters, be it carrier proteins or channel proteins, are used up totally when they reach to the saturation point. Clear till here? Clear up to facilitated diffusion? Next. So, it is the diffusion of what molecules? Hydrophilic substances. I already told you, those molecules which have hydrophilic moiety, they will be transported by channel proteins or carrier proteins. It also needs a concentration gradient, and this will be very specific. They are very specific. Cells select substances for the uptake, and I told you that these are sensitive. The channel proteins or carrier proteins, they are sensitive, which reacts with protein side chains. Okay. Transport rate will reach a maximum. Transport rate will reach maximum when all protein transporters are being used and when they reach up to saturation. Clear till here? Any doubt anywhere, ma? Now, what are the protein channels called as? Protein channels are of large sized protein channels. They are addressed as porins. Okay, they are known as porins. And those protein channels which are transporting water molecules, are known as aquaporins. Those protein channels which are transporting water molecules are addressed as what? Aquaporins. An extracellular molecule that binds to transport molecules, then it rotates and releases the molecules on the other side of the membrane. For example, water channels which are made up of 8 types of aquaporins. Water channels are made up of what? 8 types of aquaporins. Clear till here? Okay. Large sized channels are known as proteins. Those channels which are transporting water molecules are known as what? Aquaporins. Now, passive unipotes, sympotes and antipodes. Okay. When facilitated diffusion has protein channels or carrier proteins, for example, if a single molecule is being transported through the carrier protein in one direction, in a single direction, single molecule is being transported by carrier protein, that is known as what? Unipote. Okay, unipote. And if two molecules are transported parallelly by a single carrier protein, okay, or protein channel, two molecules are transported parallelly but in opposite direction. Such movement is known as what? Antipode. Okay. And sympode, when two molecules crossing the membrane in the same direction by a protein channel. Okay, two molecules are transported in the same direction, that is addressed as what? Sympode. You are clear with unipode, antipode, molecules moving in the opposite direction, sympode, molecules moving in the same direction through the channel protein. Clear till here, ma? Unipode, sympode and antipode. Now, active transport. Active transport requires energy. Okay, it is transport of molecules which is against the concentration gradient, which means from low concentration to high concentration. Low concentration to high concentration, as you see here, low to high concentration, which requires the energy, that is ATP. These are carried by some membrane proteins. These proteins are addressed as pumps. As these pumps are transporting the molecules against the concentration gradient from low to high, this is also known as what transport? Uphill transport. Remember that. This is also known as uphill transport. Transport rate reaches maximum when all the protein transporters are being used up. Same the case with facilitated diffusion, right? When all the transporters are used up, the transportation reaches the saturation level. The carrier proteins are very specific here. Carrier proteins are very specific, and these are sensitive to inhibitors. Even the case of facilitated diffusion also, the channel proteins and carrier proteins are specific and they are sensitive to inhibitors. Clear till here, ma? Any doubt anywhere? Yeah. Now, this table is very important, I am telling you, which is from your text. This table is very important. Does any membrane proteins are required? membrane proteins are required only for facilitated diffusion and active transport. Here they are addressed as what? Pumps, and here these are porins. Fine, right? Now, highly selective. Yes, carrier proteins and channel proteins, be it porins or pumps, they are highly selective for the molecules. They are highly specific for facilitated diffusion and active transport, not for diffusion. Next, transport rate reaches to a saturation level when all the transporters are used up. Okay, that happens only for facilitated diffusion and active transport again. Uphill transport that is against the concentration gradient is observed only in the active transport, not in the facilitated or symbol diffusion. Next is requirement of ATP energy. ATP energy is required only for active transport. Only for active transport ATP energy is required. It is not required for symbol diffusion or facilitated diffusion. Clear till here, ma? Any doubt anywhere? Yeah. Now we will move on to the next concept, which is plant-water relations. Okay. Before learning the plant-water relations, you have to know few concepts about water potential, pressure potential and solute potential. Okay. Remember that water is a universal solvent. Okay, water is a universal solvent, and here protoplasm or protoplast, what does it mean? If you have to consider a single cell along with cell membrane plus cytoplasm along with some cellular constituents, These are known as what? Protoplast. This is known as protoplast. What is protoplast? Cell membrane along with cytoplasm and cellular constituents is addressed as a protoplast. Fine, right? Now, what is a protoplasm? Despite the cell membrane, only cytoplasm and cellular constituents is addressed as protoplasm. Clear with the terms of protoplast and protoplasm? Now, protoplasm is mainly water in which various molecules are dissolved and suspended inside the cell. Soft plant parts may contain water. Remember, these statements are direct from your text. Watermelon has 92% of water in it, and herbaceous plants will be having 10 to 15% of water or dry matter in it. Okay, herbaceous plants have only 10 to 15% of dry matter, which means the rest percent is completely water. Dry seeds and woody parts are also containing little water. Okay, dry seeds and woody plants are also containing water. A mature corn plant absorbs 3 liters of water per day. Okay, this absorbs 3 liters of water per day. A mustard plant absorbs water equal to its own weight, about 5 hours. These are all the points that are driven from your text, and it is very important. Fine, right? These are all memory based points, and you have to be clear on the note of protoplast and protoplasm. Clear, right? Now, plant-water relationships. Here you have to know about water potential, pressure potential and solute potential. Water potential, that is nothing but potential energy or kinetic energy of water molecules, which will be maximum in the beaker containing water. Only water molecules are present in this cell, let us suppose. Only water molecules means maximum water potential will be there. Highest water potential is present here. And here if you see, this is a membrane, and here if you see, water molecules along with some solute particles are present. These solute particles are glucose molecules, and here you have low water potential. Okay. The movement of water molecules from high water potential to low water potential across a membrane is addressed as osmosis. Clear here? So, this is addressed as what? Water potential, the highest movement of water, the free movement of water or kinetic energy, potential energy of water is addressed as water potential, and it is measured in the units. Those units are what? Pascals. It is measured in pascals. Water molecules, they have high kinetic energy in liquid and gaseous form. They show random and rapid constant motion. As concentration of water in a system increases, kinetic energy or water potential also increases. Kinetic energy or water potential, it will increase. Hence, pure water here, they have greater water potential. Pure water is having greater water potential. Clear till here? Any doubts anywhere about water potential, ma? Clear, right? Now, see, look at here. Water potential of pure water will always be 0 at standard temperature and pressure. Pure water, water potential will always be 0 when they are moving to a solute particles. Here, as solute particles are present, water potential, that is movement of water, energy of water, free movement or kinetic energy of water will be decreased. Water potential from pure water to water potential in solute molecules, it will be negative. In solute or in the presence of solute potential, the water potential will be negative, and in pure water at standard temperatures, it will be 0. Clear? Next. Here we go. A solute which is dissolved in the water, here free energy of water will be decreased. Magnitude of lowering water potential is addressed as solute potential. What is solute potential? Solute potential is nothing but the magnitude by which it is reducing water potential. Okay, solute molecules will decrease the free energy of water here. The energy that solute molecules are putting to reduce the energy of water molecules is known as solute potential. Okay, the energy that is required by solute particles to reduce the free energy of water molecules is known as what? Solute potential. Magnitude of lowering water potential due to dissolution of solute is known as solute potential. Solute potential is also addressed as osmotic potential, you remember it. As solute potential will always reduce the energy of water molecules, that solute potential will always be negative, remember that. Remember that solute potential will always be negative. What is solute potential? For any solution at atmospheric pressure, water potential is directly equal to solute potential. At atmospheric pressure, water potential will be equal to solute potential, remember that. Okay, solute potential or osmotic potential, that is always negative, and at atmospheric pressure, water potential is always equal to solute potential. Clear till here? Any doubt anywhere, ma? Now, what is pressure potential? You got a clear idea on solute potential, you got a clear idea on water potential. Now, what is pressure potential? When water enters into a plant cell due to diffusion process, when water enters into a plant cell, it makes the cell turgid, right? This increases the pressure potential as it is. For example, this is a cell wall, and let us suppose this is the cell membrane inside which molecules are present, and water has entered here. Here water potential is decreased and water molecules are being added here completely, which is increasing the cell membrane size, and here cell membrane is exerting some pressure on the cell wall, right? This pressure by which it is putting on the cell wall, okay, the pressure that is kept by cell membrane on the cell wall as the cell is getting turgid, okay, that potential is known as pressure potential. When water enters into a plant cell due to diffusion, this causes a pressure against the cell wall. Against the cell wall, some pressure is created. This will make the cell turgid on hole. This increase in the pressure is known as pressure potential. Fine, right? Any doubt anywhere? Clear till here? Now, pressure potential is usually positive. Remember that. Pressure potential is usually positive. Though negative potential or tension in the water column in the xylem plays major role for the transport all across the stem. Fine, right? When there is more solute concentration in the stem, only the force will be driven in the xylem tissues for the movement of water to the tip of the plant. So here, how you can calculate water potential on hole? Water potential or the movement of water is calculated by solute potential along with pressure potential will give you out the water potential. Okay, water potential is equal to solute potential along with pressure potential. Clear till here? Any doubt anywhere, ma? Now, osmosis. It is spontaneous diffusion of water across differentially or semi-permeable membrane. Okay, spontaneous diffusion of water across differentially or semi-permeable membrane. Here cell membrane or tonoplast. Okay, cell membrane or tonoplast, which is the membrane of vacuole, are important determinants for movement of water molecules in and out of the cell, but cell wall is not a barrier for the movement. Cell wall is completely permeable for the movement of water and the substances of in the solution. Fine, right? Vacuolar sap here. The molecules which are present inside the vacuole are addressed as what? Vacuolar sap. In large central vacuole, this will contribute to solute potential of the cell. Clear till here? Any doubt anywhere, ma? Yeah. Now, osmosis. This is from high water potential to low water potential. High water potential, that is high solute potential to low solute potential. Net direction or rate of osmosis will always dependent on pressure gradient and concentration gradient. Remember that. So, the net direction or rate of osmosis is always dependent on pressure gradient and concentration gradient. Here water will move from the region of high concentration, high water potential to low water potential, that we already know. And this diagram based question has been put multiple times in your exam. Multiple times you got this question in your exam. Fine, right? Next. Potato osmometer. This is one of the experiment that you have to know. Potato, which is, take a potato and a cavity is made in the potato. Some sugar solution is added and it is pinned. To which extent the sugar solution is added, that is being pinned like this. Okay. Okay, inside the potato the sugar solution is present and it is pinned. And this potato, to which sugar solution is kept in the cavity and it is pinned, that potato is kept into a beaker containing water. Okay. Now obviously in the osmosis process what happens? The water molecule will move from the high water potential to low water potential. High water potential is present in the water containing beaker and it is moved into the sugar solution which is present inside the cavity. Fine, right? This is from high water potential to low water potential. Fine, right? So after osmosis, if you see another pin marked over here, this much level of rise in water is seen in the cavity of the potato. This is potato osmometer. This complete setup is addressed as what? Potato osmometer, to check the rise in water before osmosis and after osmosis, and to determine and to say that water, the osmosis is always from the high water potential to low water potential. Fine, right? Clear till here? Everything is fine? Now, here testil funnel which is filled with sucrose, same experiment But here you will be coming across two terms, one is osmotic pressure and pressure potential you already know. Osmotic pressure. So, in this setup what happens is a distal funnel into which sugar solution is placed, above which the egg membrane is covered, which is semi-permeable membrane, and this funnel is inverted into a water, into a beaker containing water. Fine, right? Now, obviously, the molecules will be moved from the beaker containing water into the distal funnel. Fine, right? And the pressure is exerted over this for the movement not to happen from the beaker to the distal funnel. The amount of pressure that is exerted here to reduce the flow of water movement from beaker to the distal funnel is addressed as what? Osmotic pressure. In simple terms, the pressure which is required to stop the process of osmosis or to reduce the process of osmosis is addressed as what? Osmotic pressure. Fine, right? So, this will demonstrate osmosis and osmotic pressure. Here you can go through. If an external pressure is applied from upper part of the funnel, no water will diffuse into the funnel. Fine, right? This pressure is required to prevent the water diffusion. This is addressed as what? Osmotic pressure. This is function of solute concentration. More than the solute concentration, greater will be the pressure required to prevent the water diffusion. Numerically, osmotic pressure is equivalent to what? Osmotic potential, but the sign is always opposite. Osmotic pressure is positive, and osmotic potential is always negative. Remember that. Clear? Now, plasmolysis. Here there are three experiments that are shown. A resin or any cell. Let us assume a resin which is placed into a high concentration solution or high sugar concentration solution, concentrated solution. Fine, right? Okay. The water will always move from high potential to low potential. So, it will be from resin to the outer environment. And here the cell membrane will shrink from the cell wall. Any cell which is placed into a hypertonic solution with high solute molecules, the water molecules will move from the cell to the solution, which will make the cell shrink from the cell wall. This cell is addressed as a plasmolyzed cell. Okay. And turgid cell, if you see, when a cell is placed in a hypotonic solution, here the water molecules from the solution which is of high water potential, they will move into the cell, which will make the cell turgid. It will increase the pressure potential, it will increase the cell membrane's pressure on the cell wall, turgor pressure will be increased and it will make the cell turgid. Clear? Now, if an external solution balances osmotic pressure, it is called as isotonic cell. There is no net flow of water there, the cell will be in equilibrium in the second case, which is addressed as a flaccid cell, where water molecule will move in and out, this stays in an equilibrium. I hope the three cases are clear to you, both hypertonic solution, hypotonic solution and equilibrium. Fine. Now, so here you can see microscopic view before plasmolysis and once a cell is plasmolyzed, how cell is shrinked away from the cell wall. Clear? Now, about turgor pressure and pressure potential. Turgor pressure or pressure potential can be used as synonyms. Turgor pressure or pressure potential can be used as what? Synonyms. Because, listen here, if this is a the cell wall and cell membrane. Fine, right? When water molecules are entering into the cell and making the cell turgid or increase its cell size, here a pressure is exerted by the cell membrane towards the cell wall. Okay. This pressure by which the cell membrane is exerting on the cell wall is addressed as pressure potential. Pressure potential or turgor pressure. Clear till here? Any doubt anywhere? Now, the last concept for today, that is imbibition. Okay. Many a times we have observed during rainy season, the size of wood will be increased. That is because of imbibition, we will say. What does that actually mean? Okay. The increase in the size of solid because of entry of water into the cells is known as imbibition. Okay, which is nothing but absorption or diffusion of water into solid particles. Okay, diffusion of water molecules into the solid particles is known as what? Imbibition. Diffusion of water into solids or colloids is addressed as imbibition. For example, absorption of water by seeds and dry wood. The pressure is due to swelling of wood and this can even split the rocks. Seedlings are emerged out of the soil, this is due to imbibition pressure. Seedlings are emerged out of the soil, that is due to imbibition pressure, that is water molecules entering into the hard and hard coated seed. Imbibition will require few conditions, few factors will affect the process of imbibition here. What are those factors? Difference in the concentration gradient, difference in the concentration gradient. Water potential gradient is in between the absorbent and liquid that is imbibed. This is in between the absorbent and the liquid which is imbibed. Affinity between adsorbent and liquid is high. Affinity between adsorbent and liquid is present over here. Clear? Any doubt anywhere? Now, tomorrow we will look into long distance transport and we will exercise EAPCET PYQs on the transport in plants on whole. So, today what are the concepts that we have covered, ma? First thing, active and passive transport, fine, right? Active and passive transport, where in passive transport we have clearly dealt with diffusion, facilitated diffusion and osmosis. Fine. What are the factors that are affecting diffusion, facilitated diffusion and osmosis? This we have dealt clearly in a tabulated form, which is more specific and where porins are required, what are pumps, what is passive transporters also we have discussed, uniport, symport and antiport. Fine, right. After which we have moved on to plant water relations, where we have dealt in clear with water potential, pressure potential and solute potential. And then we have looked on to few experiments like potato osmometer and to derive osmotic pressure and osmotic potential. And lastly, we have gone through two major processes, one is plasmolysis and second is imbibition. These concepts are the major concepts both which are appeared many a times. If you see regarding plasmolysis also, many a times the application based questions are asked on plasmolysis. On imbibition also many a times the questions are put. In our next class we will move on to long distance relations, okay, where we will completely analyze the transport of xylem and the transport of phloem, both unidirectional and multidirectional transport. In today's class we have got the gist of unidirectional and multidirectional transport. In our next class we will have a clear view on what is unidirectional transport and multidirectional transport on whole, and we will also learn apoplastic pathway and symplastic pathway by once again recalling the anatomy of root. So, till here everything is fine, Oma? That's all for today's session, and in next class we will look into all the long water distance transports. Fine, right? Yeah, that's all for today's session, and thank you.